Welcome, medical students, to today's lecture on mechanisms of DNA replication errors and mutagenesis. We will explore DNA replication errors, mutagenesis rates, chemical carcinogens, cancer development, high-throughput sequencing, structural insights, hereditary diseases, Ames test, regulatory toxicology, and emerging trends in mutagenesis research. Let's begin. DNA replication is a process where genetic material is duplicated accurately within cells. However, mistakes can occur resulting in mutations in the DNA sequence. These errors can come from DNA polymerase mistakes, DNA damage, or misincorporation of nucleotides. Environmental factors like radiation or chemicals can also cause DNA structural damage. These mistakes can lead to genetic diseases or cancer. Cells have evolved complex DNA repair pathways to detect and correct DNA damage, maintaining genomic integrity. These pathways include base excision repair, nucleotide excision repair, mismatch repair, and double-strand break repair. Each pathway recognizes and repairs specific types of DNA lesions, preventing mutations. Understanding these systems helps preserve genome integrity. Chemical carcinogens are substances that can cause cancer by damaging DNA. They can be found in industrial byproducts, tobacco smoke, and certain foods. These substances can directly bind to DNA, causing structural changes that lead to mutations during DNA replication. Some chemical carcinogens can also produce reactive oxygen species, which directly damage DNA and contribute to mutagenesis. Understanding the mutagenic properties of chemical carcinogens is important for assessing their risks and developing prevention strategies. In cancer development, genomic instability and mutagenesis are critical factors. Genomic instability, characterized by increased DNA mutations, can stem from defects in DNA repair, abnormal cell cycle control, and exposure to genotoxic agents. This leads to mutations in genes involved in cell growth control, DNA repair, and tumor suppression, ultimately causing uncontrolled cell growth and cancer. Understanding this relationship is crucial for developing targeted cancer therapies. Advancements in sequencing technologies have transformed our ability to study mutagenesis on a whole genome level. High-throughput sequencing, like next-generation sequencing, enables efficient and rapid analysis of massive DNA sequence data. These technologies help identify mutational patterns and understand the causes of mutagenesis. By examining mutation patterns across the genome, researchers can identify the effects of different mutagens, like UV radiation or specific chemical carcinogens. Additionally, high-throughput sequencing aids in identifying driver mutations that contribute to cancer development, paving the way for personalized therapies. The application of these technologies has revolutionized our understanding of mutagenesis in cancer research and precision medicine. In this video, we will explore how understanding the structure of DNA and proteins involved in mutagenesis helps us understand how mutations occur and how they can be repaired. By studying the interactions between DNA and proteins, we can learn more about the factors that influence accurate DNA replication. We will also see how structural studies have provided insights into the mechanisms of DNA repair proteins. By combining structural biology with other research techniques, we can gain a better understanding of mutagenesis and potentially develop new therapies. Mutations in germline DNA can cause hereditary diseases, which may result in metabolic disorders, developmental abnormalities, or increased cancer risk. Understanding mutagenesis is crucial for diagnosis, early intervention, and genetic counseling. It also enhances our knowledge of DNA replication errors. The Ames test is used to assess the mutagenic potential of chemicals. It measures the ability of a chemical or drug to revert mutations in bacterial strains, indicating potential DNA mutagenesis. Standardizing the test methodology ensures reliable results. In vitro mutagenesis models are tools that help us study how chemicals can cause mutations. They allow us to control experiments and study mutations in a lab. One model uses cell cultures to test mutagenicity, while another uses recombinant DNA technology to intentionally introduce mutations. These models help us understand mutations and develop strategies to mitigate their harmful effects. The Ames test is a critical tool in regulatory toxicology, used to quickly and inexpensively assess the mutagenic potential of chemicals. It helps identify potential mutagens and prioritize further testing, aiding in the prevention of human exposure to harmful compounds. The test also helps establish structure-activity relationships, aiding in the prediction of mutagenicity for similar compounds. This inclusion of reliable mutagenicity assays, such as the Ames test, in regulatory toxicology workflows helps safeguard human health and the environment. Chemical risk assessment helps evaluate hazards and risks of substances. Mutagenicity assessment is crucial in assessing risks to human health. The Ames test detects DNA mutagenesis in bacteria, aiding in assessing mutagenic potential. Combining Ames test results with other toxicological data allows informed decisions on regulation and safe handling of mutagenic compounds. This enhances public safety. Summarize in 33 words. Mutagenesis research is evolving with genomics and bioinformatics integration. High-throughput sequencing generates data to identify mutational signatures. Bioinformatics tools analyze complex datasets, extracting insights. Advanced genetic engineering like CRISPR-Cas9 helps study mutations and their functional consequences, guiding prevention and therapeutic strategies for diseases. In summary, knowing how errors in DNA replication lead to mutations is vital for understanding genetic diseases and cancer. The Ames test is a reliable way to measure mutagenicity. Advancements in genomics and genetic engineering will further our understanding of mutagenesis and help prevent diseases.